Today we will be talking about virus. A virus is a tiny infectious agent that reproduces inside the cells of living hosts. When infected, the host cell is forced to rapidly produce thousands of identical copies of the original virus. Unlike most living things, viruses do not have cells that divide. New viruses assemble in the infected host cell. Viruses contain genes, which allow them to mutate and evolve. Over 4,800 species of viruses have been described in detail out of the millions in the environment. Their origin is unclear. Some may have evolved from plasmids. It is pieces of DNA that can move between cells. While others may have evolved from bacteria. Viruses are made of either 2 or 3 parts. All include genes. These genes contain the encoded biological information of the virus and are built from either DNA or RNA. All viruses are also covered with a protein coat to protect the genes. Some viruses may also have an envelope fat-like substance that covers the protein coat and makes them vulnerable to soap. A virus with this viral envelope uses it along with specific receptors to enter a new host cell. Viruses vary in shape from the simple helical and icosahedral to more complex structures. Viruses range in size from 20 to 300 nanometers. It would take 33,000 to 500,000 of them, side by side, to stretch to 1 centimeter, 0.4 in. Viruses spread in many ways. Some viruses of humans and other animals are spread by exposure to infected bodily fluids. Viruses such as influenza are spread through the air by droplets of moisture when people cough or sneeze. Viruses such as norovirus are transmitted by the fecal, oral route, which involves the contamination of hands, food, and water. Rotavirus is often spread by direct contact with infected children. The human immunodeficiency virus, HIV, is transmitted by bodily fluids transferred during sex. Others, such as the dengue virus, are spread by blood-sucking insects. Viruses, especially those made of RNA, can mutate rapidly to give rise to new types. Hosts may have little protection against such new forms. Influenza virus, for example, changes often, so a new vaccine is needed each year. Major changes can cause pandemics, as in the 2009 swine influenza that spread to most countries. Often, these mutations take place when the virus has first infected other animal hosts. Some examples of such zoonotic diseases include coronavirus in bats and influenza in pigs and birds, before those viruses were transferred to humans. Viral infections can cause disease in humans, animals and plants. In healthy humans and animals, infections are usually eliminated by the immune system, which can provide lifetime immunity to the host for that virus. Antibiotics, which work against bacteria, have no impact. But antiviral drugs can treat life-threatening infections. Those vaccines that produce lifelong immunity can prevent some infections. The invention of the electron microscope in 1931 brought the first images of viruses. Viruses are among the smallest infectious agents and are too small to be seen by light microscopy. Most of them can only be seen by electron microscopy. They range in size from 20 to 300 nanometers. In contrast, bacteria are usually about 1000 nanometers, 1 micron, in diameter. And host cells of advanced organisms are usually dozens of microns. Some viruses, such as the Big virus and Pandora virus, are relatively large viruses. These viruses were found in 2003 and 2013 respectively. They are about 10 times wider than the flu virus and therefore 1,000 times larger. The discovery of these giant viruses surprised scientists. Genes. The genes of viruses are made from DNA, deoxyribonucleic acid, and, in many viruses, RNA, ribonucleic acid. The biological information contained in an organism is encoded in its DNA or RNA. Most organisms use DNA. But many viruses have RNA as their genetic material. The DNA or RNA of viruses consists of either a single strand or a double helix. Viruses can reproduce rapidly because they have relatively few genes. For example, influenza virus has only 8 genes and rotavirus has 11. In comparison, humans have 20,000 to 25,000. Some viral genes contain the code to make the structural proteins that form the virus particle. Other genes make non-structural proteins found only in the cells the virus infects. All cells, and many viruses, produce proteins that are enzymes that drive chemical reactions. Some of these enzymes, called DNA polymerase and RNA polymerase, make new copies of DNA and RNA. A virus's polymerase enzymes are often much more efficient at making DNA and RNA than the equivalent enzymes of the host cells. But viral RNA polymerase enzymes are error-prone, causing RNA viruses to mutate and form new strains. In some species of RNA virus, the genes are not on a continuous molecule of RNA, but are separated. The influenza virus, for example, has 8 separate genes made of RNA. When 2 different strains of influenza virus infect the same cell, these genes can mix and produce new strains of the virus in a process called reassortment. Protein synthesis. Proteins are essential to life. Cells produce new protein molecules from amino acid building blocks based on information coded in DNA. Each type of protein is a specialist that usually only performs one function. So if a cell needs to do something new, it must make a new protein. Viruses force the cell to make new proteins that the cell does not need, but are needed for the virus to reproduce. Protein synthesis consists of 2 major steps: transcription and translation. Transcription is the process where information in DNA, called the genetic code, is used to produce RNA copies called messenger RNA, mRNA. These migrate through the cell and carry the code to ribosomes, where it is used to make proteins. This is called translation. Because the protein's amino acid structure is determined by the mRNA's code, information is hence translated from the language of nucleic acids to the language of amino acids. Life cycle. When a virus infects a cell, the virus forces it to make thousands more viruses. It does this by making the cell copy the virus's DNA or RNA, making viral proteins, which all assemble to form new virus particles. Effects on the host cell. Viruses have an extensive range of structural and biochemical effects on the host cell. Most virus infections eventually result in the death of the host cell. The causes of death include cell lysis, bursting, alterations to the cell's surface membrane, and apoptosis, cell suicide. Often cell death is caused by cessation of its normal activity due to proteins produced by the virus, not all of which are components of the virus particle. Some viruses cause no apparent changes to the infected cell. Cells in which the virus is latent, inactive, show few signs of infection and often function normally. This causes persistent infections and the virus is often dormant for many months or years. This is often the case with herpes viruses. This is the basic knowledge about virus. Thanks for watching. If you like this video, please thumb up and leave a message. You must be my biggest motivation to make the video. Please subscribe to the channel, where you'll find more and more medical videos here later. Thanks.